Clostridium botulinum is a rod-shaped, gram-positive anaerobic spore-forming bacterium recognized for its production of botulism toxin. This toxin can lead to severe foodborne illness, termed botulism, and characterized by muscle paralysis, respiratory failure, and in some cases, mortality. Clostridium botulinum's endospores are highly resistant to environmental stresses such as high heat and low acidity. They can remain dormant for extended periods of time until favorable conditions for growth are present. When conditions improve, the spores germinate and produce vegetative cells that produce a potent neurotoxin known as botulism toxin. Clostridial botulinum spores are found everywhere in nature, commonly located in soil, water, sediment, and the intestines of various animals. These spores tend to thrive in low oxygen environments, which facilitate their growth and toxin production. Botulism bacterium may contaminate a diverse range of food products, particularly those that have been improperly canned or preserved. Foods that commonly present a heightened risk include home canned goods, especially vegetables and meats, as well as fermented fish and select fruits that have not been adequately prepared and or processed. The introduction of Clostridium botulinum into the food supply presents a significant public health concern that the food safety experts must address with urgency and diligence. This bacterium, found in soil and untreated water, produces a highly potent neurotoxin that can cause botulism, a rare but potentially fatal illness, characterized by muscle paralysis and respiratory failure. The risk of botulism is particularly heightened in improperly canned or preserved foods, where anaerobic conditions allow the bacteria to thrive and produce toxins. Factors such as inadequate heat treatment, failure to maintain appropriate acidity levels, and improper storage temperatures can contribute to the proliferation of botulism. To combat this threat, food manufacturers and processors must adopt stringent safety protocols throughout the production and distribution process. This includes employing validated canning techniques that ensure complete sterilization and utilizing high-temperature pasteurization for low-acid foods. Regular monitoring of pH levels and moisture content is also crucial to inhibit bacterial growth. Furthermore, implementing HACCP system that can identify potential hazards and establish critical control points to mitigate risks effectively. Clostridial botulinum exhibits optimal growth at temperatures ranging from 30 degrees Celsius to 40 degrees Celsius, so approximately 86 degrees Fahrenheit to 104 degrees Fahrenheit. This temperature range is particularly concerning for improperly stored or processed foods. The bacterium favors a pH of 4.6 or lower for optimal toxin production. However, its spores are capable of surviving a broader pH range, presenting significant challenges to food safety, particularly in low-acid foods. Clostridium botulinum possesses the ability to form highly resistant spores that can endure extreme environmental conditions, including heat and desiccation. These spores can survive standard cooking temperatures and may remain dormant for significant periods only to germinate and produce toxins when conditions become favorable. Toxin production primarily occurs under anaerobic conditions, particularly in low acidic environments. Foods that are improperly stored or sealed can create ideal conditions for this process, highlighting the necessity for food processors to adhere to established safety protocols. Canning is a method of food preservation that involves placing food in jars or cans, sealing them, and heating them to kill harmful microorganisms and inactivate enzymes that can lead to spoilage. Typically, high-acid foods such as fruits and pickles are processed at boiling water temperatures of 212 degrees Fahrenheit or 100 degrees Celsius for a specific duration to create a vacuum seal that prevents air from entering. This vacuum is crucial for inhibiting the growth of Clostridium botulinum. Retorting, on the other hand, is a specialized form of canning that uses pressure cooker or retort to process low-acid foods, such as vegetables and meats. The retorting process involves heating sealed containers to temperatures exceeding 240 degrees Fahrenheit. Under pressure, typically at around 10 to 15 psi. This elevated temperature is necessary to destroy the spores that can survive in low-acid environments. Retorting ensures that these foods are safely preserved while maintaining the quality and nutritional value. Both canning and retorting aim to preserve food and prevent botulism by using heat treatments to eliminate harmful bacteria. They create airtight seals to keep oxygen out, which is essential for inhibiting bacterial growth. The key difference lies in the type of food processed and the methods used. Canning is typical for high-acid foods and uses boiling water at 212 degrees Fahrenheit, while retorting is for low-acid foods and employs high pressure to reach temperatures of 240 degrees or higher. This difference in temperature and pressure is crucial for ensuring food safety and quality. Effective food safety management requires the identification of hazards associated with Clostridium botulinum, focusing on both the presence of spores and the potential for toxin production in a variety of food products. Critical control points in food processing sequence must be determined where control measures can be implemented. For example, cooking temperatures, pH monitoring, and critical limits. To be more specific, ensuring that retorting achieves the necessary temperature to effectively eliminate spores, with regular assessments of the pH levels in canned products are essential to ensure they remain within safe parameters. And finally, it is vital to establish specific thresholds, such as maintaining a temperature of 121 degrees Celsius to guarantee the safety of food products. Advantages of food processing are simple. Food processing significantly extends the shelf life of products by inhibiting microbial growth and thusly reducing the overall microbial load. When executed correctly, food processing can preserve the nutritional integrity of foods, ensuring consumers receive quality products that retain essential vitamins and minerals. Disadvantages of food processing are also simple. Improper processing practices can lead to serious health risks, including foodborne illnesses from pathogens such as Clostridium botulinum, if safety protocols are not strictly followed. There exists potential for nutrient loss during processing, especially in methods involving high temperature or prolonged storage. Processed foods may contain preservatives or additives that some consumers prevent to avoid, thus impacting purchasing decisions. Clostridium botulinum is a significant concern for various food products, particularly home canned vegetables such as green beans and carrots, which can harbor spores if not processed correctly. Fermented fish, like certain traditional fish products, can also pose a risk if not prepared and stored properly, as the low-acid environment can support the growth of bacteria. To prevent botulism, consumers should take several important steps first. They should follow tested and reliable canning recipes, ensuring that the proper technique and equipment, such as pressure canners for low-acid foods. Additionally, consumers should inspect canned goods for any signs of spoilage, such as bulging lids and/or off odors. Lastly, consumers should avoid any compromised products. Proper storage of fermented fish and other low-acid foods at safe temperatures, along with thorough cooking before consumption, can significantly reduce the risk of botulism. Understanding Clostridium botulinum is not only vital for food safety, but also imperative for public health. The bacterium possesses significant risk, particularly in low-acid foods that are improperly stored or preserved. Education on the nature of this organism and its toxins can empower both food processors and consumers to take proactive steps in preventing foodborne illness. Important food safety practices must prioritize the prevention of botulism include proper canning techniques, monitoring pH levels, awareness of symptoms, safe cooking practices, role of food industry, and regulations. In conclusion, the threat posed by Clostridium botulinum underscores the importance of food safety education for both processors and consumers. By implementing safe canning and cooking practices, monitoring pH levels, and recognizing the sign of botulism, we can substantially mitigate the risk associated with this dangerous bacterium. A collaborative effort between the food industry, regulatory agencies, and consumers is essential in ensuring the safety and quality of our food supply, ultimately protecting public health.